there's a tremendous number of questions, then then we could stay longer. But I don't. With this group, I don't anticipate that's going to be the scenario. And then for those of you who would like to actually use these models, tomorrow we'll have a hands-on tutorial using a coarse-grained model, and on Friday we'll be using the all-atom model and talk about some new tools that we've developed that can be useful when you're trying to apply these models to various systems, and what are some considerations for performance and related topics. Right, so in my talk and other talks, you've heard people talk about these structure-based models. So we'll just go through, what they are. I'll first give thanks to a couple of postdocs that have helped put these slides together. This little mini workshop, something that stemmed from something Jeff and I did about maybe 8 years ago, because there were experimental students who went to talks and they would see results, but they never knew what they meant, and they didn't really have a feel for where the results came from. They would just see a free energy profile. And they said, like, that looks pretty, but we really have no grasp of what's happening. So we started organizing these through the Center for, through CTPP over at San Diego, and then through the years we've had a variety of additional workshops. And these two guys, Chris and Jeff, have contributed pieces. So I always like to thank them, even though they're not here. So as I said, it's a basic discussion of how are these models formulated, what are the terms, what goes into them. They're really not that complicated, but people often feel there's some sort of mystery, there's something hidden, and that's not the case. So we'll go over that today. What are some, whoops. Oh, I didn't realize you plugged that in. Okay. And there. Okay, and what are some options, some considerations, or ways you could apply these models? So tomorrow we'll actually go through an example calculation, set up one of these models, you'll see how the tools work. And then on Friday, we'll go through a larger scale system with a higher resolution model and consider performance considerations. So yeah, like I said, I didn't know exactly who was going to be here, so the first thing I wanted everyone to do, there's maybe too many people, was to, and I know most of you, but anyway, was for you to tell me what your background is so I could tailor the workshop to the audience. I don't think that's really useful right now. So I think most of you have used structure-based models, but just by a show by hands, how many of you have actually used a structure-based model in any capacity for a calculation before? This is useful for me, so for the next couple of days I have a feel. Yes, raise your hand so I know if it's like everybody or just sort of a sprinkling. So about half of the audience. Okay. So people who have used these models probably know how they're constructed. And if you want to participate in actual exercises in the next 2 days, there are several software packages that you should install. Visual Molecular Dynamics, or VMD, GROMACS version 5. These are free downloadable software packages. And ideally, you'll download our structure-based model software as well, SMOG version 2. You don't need SMOG 2 to configure completely. We're only going to be using the tools aspect, so it's a little simpler. So as I said, there's sort of a history behind these models, and I like to explain that history to people who are coming in from the outside. A lot of you actually have been involved in these various steps through the years, so feel free to chime in and add additional information as we go. So they really stem from the 80s and 90s of these ideas of unfrustrated energy landscapes, and they started with lattice models, moved on to coarse-grained models. In the 2000s, this is sort of where I came in. We pushed these all-atom versions of the models. And now we have this class of models that can handle coarse graining, all-atom resolution, proteins, RNA, DNA, ligands, you name it, you can actually apply the concepts. I'll show you, yeah. and then I'll give a couple of highlights of some applications of these models that people have used that are not extremely demanding applications, but it might spur some ideas of ways you could use them or integrate them into your work. So a lattice model is exactly what the name you would think from the name, it's basically a spin lattice system, but instead of having indistinguishable spins, the spins are distinguishable, so you have occupancy on the lattice, and the spins are coupled to one another in space. So you have a chain. That's, from a model perspective, it's no different than an Ising model with constraints on the spins. So spin ones have to be next to spin ones, and they have to be in order. And so back in the 80s, there were a lot of groups in 80s and 90s, people were working with these types of models, and these aren't meant to be, you know, have everything, but they include some basic elements of proteins, such as chains can't cross themselves. Right? There's a finite length to the chain. There are more unfolded configurations or extended configurations than compact configurations. And with this representation here, this is just a few colors representing different energetic parameters. So you could say maybe hydrophobic residues are just attractive orange beads. Charged residues could be blue and red, and you can assign energetics, so it's a slightly more complicated spin model. Right? So the coupling constants actually depend on species. And so with these models, actually Whitford did a lot of the work on these models in the past. There were a variety of basic physical questions of chain length effects, diffusion effects, say looking at the balance between short-range and long-range interactions, because even though it's a spin model, you could have only adjacent spins interact, or you could have spins to some arbitrary length scale interact. And you could address these sort of at a very crude level, balance of energetic distributions and cooperativity of folding, for example. And what was nice about these models was that they were very cheap. You could basically enumerate all possibilities. So when computers were a little less powerful 20, 30 years ago, that was very, very helpful. Nowadays, these models have gone somewhat out of fashion, but the ideas behind the new models are the same. So with those lattice models, you could say that only it's a representative of sort of an ideal protein. It's not a real protein because they're not straight lines and 90-degree turns in proteins. But you can encode the interactions such that they sort of mimic the general structure of a protein, of a compact globular thing. And, Vitor, chime in. What did people learn from the lattice models? He was really involved back then. Oh, I didn't see Husan back there. Husan, see, so what were some of the exciting things that you learned when using these lattice models back? So what are the minimal elements that you need to fold a protein? So that's, yeah, this is great. I have people who actually worked. I've never worked with these models, so I can ask people who did. They've done a lot with these models. So, but that was the general idea. Is it brought this idea of these unfrustrated landscapes where you have native interactions being dominant, and they stabilize the folded configuration, and that gave rise to this funnel sort of idea where you have a large energy gap between folded and unfolded ensembles, and the roughness is small. So let me see. This pointer is a little weak. Okay. And with the lattice models also, you could just enumerate all of these states roughly. In 3 dimensions, it gets to be a little large, but in 2 dimensions, they knew every possible configuration that could exist, so evaluating the partition functions was literally just doing a sum. Right? Getting all the thermodynamic parameters was a sum. And so that's the principal idea behind all these models, is that these landscapes must lack large-scale energetic roughness, and there's a dominant configuration that where the interactions are effectively stabilized. So people were using those approaches, and then around 2000, people start looking at these off-lattice models, which is what we would call everything else. Right? Anything that just moves in space is off-lattice. Right? So instead of having Monte Carlo searches on a lattice, you have integrating Newton's equations of motion, and you just update the positions, update the velocities, and you integrate the equations of motion in time. So with these coarse-grained structure-based models, you do the same thing. Now you can take actual experimental structures. So you don't need to map a protein onto a lattice, which is imperfect, but you can learn a lot. But you can actually just take PDB structures and coarse-grain it directly, and then define a force field based on that structure. And again, I'll give the force field in a second, but the structural information is directly encoded in the model. So you can't make a structure-based model for one protein and then apply it to a different protein. Every force field is specific to that particular system that you are simulating. So they're completely non-transferable. They're completely specific to that system. And that's very different from all-atom explicit solvent simulations, where you put in all the partial charges, put in all the van der Waals terms, put in the water, and then you just let it go, and you see where's the minimum. Here, we're pre-assigning it. And in these early models, a coarse-grained representation was used, where instead of representing every atom, you only represent some of them. So again, you start with a crystal structure or an NMR structure. This would be just a representation in sticks, or space-filling representation, and then you have to decide what representation you want to use exactly. Even when you say coarse-grain, there are lots of coarse-grain model variants available. So the one that we'll be talking about tomorrow are these C-alpha structure-based models. So the protein is represented as a collection of beads, where a bead is positioned at the position of the C-alpha atom. And that's just the most basic implementation of this model. You could put the bead at the center of mass of the residue, for example. You could have multiple beads per residue, so have one for the side chain, one for the backbone. It's all flexible how you want to construct them. But this particular implementation is going to be just one bead per residue. And after you define your resolution, you simply define all of the interactions to be stabilizing that particular configuration. So first you have your covalent geometry that must be maintained. You have harmonic terms for bonds, angles, your improper dihedrals if you need them. So if you're excluding hydrogen, you have chirality, but that can be lost if you don't include an improper dihedral to maintain chirality of the remaining 3 atoms. And we just put those in as strong covalent, strong harmonic terms, because covalent geometry should be maintained pretty stringently in whatever model that you would like to look at. And all of these distances, R naught, theta naught, chi naught, are all taken from the structure that you put in. And this is a condition that you can relax if you want. But again, the most basic formulation is you take the structure and you define it completely by that structure. You could put in variations where you put in distances or angles based on the sequence if you want it to, but then it wouldn't be a purely structure-based model. It would be a sort of a variant. And then if you're looking at protein folding or any sort of dynamics, the dihedral terms and the contact terms are what really drive the long time scale dynamics of the system. So for dihedrals, we just put in a sum of cosine terms. And for contacts, basically you have some algorithm to identify which two atoms are near each other, and this could be based on just a cutoff condition. It could be based on surface complementarity rules. There are many techniques that are out there. We have a variety of available options that we've tested in the context of protein folding, RNA folding, and so on. But essentially, you're just trying to say which atoms are near each other in this configuration. And if they're near each other, you give it some favorable interaction. And this could be an interaction that is of a 6-12 form or a 10-12 form. It could also be a Gaussian potential if you prefer. There are a lot of specifics, but you would always define that minimum to be at the distance that those two residues have in that configuration that you're starting with, that you're using to define the model. And then finally, you have a repulsive term or just an excluded volume term to make sure your chain doesn't cross itself. Okay? So you can see this isn't a huge departure from the lattice model, except that the atoms are allowed to move freely in space as opposed to being restrained or constrained to a lattice. Okay? But the elements are the same. And in all of these terms, these are all defined by the input structure. Okay? So if you use a bad crystal structure, you're going to get a bad force field, so you should really think about what structures you're using. You can certainly find crystallographic structures or NMR structures that have errors, so you want to look closely at your headers, make sure they haven't said there's a problem in this region, then you build a force field based on a region that they don't even believe is correct. Right? They think there's a problem there. So those problems will be carried on into the force field if you're not carefully looking at the structure you're using for defining your model. So oftentimes when you want to look at your contacts, we look at something called a contact map, and this is just residue number and residue number. And if there's a dot, it means that you've identified those two residues as interacting in that input configuration. Okay? And this, again, there are a variety of ways of constructing this map, and for those of you who are really interested, I can give you a number of papers that have compared many different definitions and the effects it can have on protein folding, for example, but I'm not going to go into much detail about how it's constructed. But when you look at this, you can start to see different features, such as along these diagonals, those are alpha helices because you have an I and I+3 and I+4 contacts that are detected because of that helical structure. These off-diagonal regions are typically anti-parallel beta sheets that appear, and then some of this could just be more sort of a globular region that isn't necessarily specifically a helix with a helix or a beta sheet. So you get a lot of long-range interactions that can come in as well. So we're starting with the C-alpha model. So in the C-alpha model, we actually don't have impropers or planars because we only have one bead per residue. And the side chains are not accounted for explicitly. The size of each side chain can be implicitly accounted for in the contacts. That is, if you have two side chains interacting and you put their C-alpha minimum at a certain distance based on those two side chains interacting, there will effectively be a repulsion if you get too close. But it's not accounted for explicitly with the C-alpha model. These are all just taken directly from the structure. So whatever three consecutive beads, whatever angle they form, you define theta naught to be that angle. Again, there is no, in the C-alpha model, this term actually isn't here because there are no improper dihedrals or planar dihedrals because it's only one bead per residue. Mm-hmm. Mm-hmm. So, right, so sorry, this notation, I need to improve. Right, I need to improve this notation. This should be R naught I, and this would be Ri. So over all adjacent residues, you have an Ri, or a distance between them, and an Ri naught would be the distance between those two residues in the structure. Yeah, so sorry, everything here should have a sub I. Right, what? It depends on how you count. If you're just giving a list of dihedrals, you could list them with I. But yes, it could be IJ, IJK. Yep. It depends on the structure, right? So it does. By default, it does. So the default structure-based model that is supported by our web tool that we'll use, that sets every R naught to be the value of those two atoms in that structure. And so each R1, R2, R1 naught, R2 naught, R3 naught can all be different. You can relax that condition with our newer software, SMOG2, if you want it to use non-specific values where everything has the same distance, for example. You can actually enable those options. So you have theta is for 3 beads, 3 consecutive beads, 1 angle, and then you have the next 3. Right, and that's what the dihedral terms are, but I haven't given a functional form. Right, this would be the torsion term. And that's also defined to have a minimum where every torsion takes the value found in that structure. Right, so the model is chiral from the dihedrals. Any other questions? Okay, so I'm describing the most standard structure-based model, but every piece of this can be relaxed. You can make it as non-specific as you like. You just have to decide how you would like to do that and for what purpose. What is the question you'd like to address by relaxing these rules? So when people started using these proteins, then this is what we're really used to seeing now. We look at, say, this is the number of native contacts to describe how folded a protein is, and you have that the protein is folded, unfolded, refolded, and you get, for many proteins that are known to be two-state folders, you still get two-state dynamics. So this model, despite its relative simplicity, it does capture a pretty major property of protein folding that would be two-state dynamics. Also, if you look at the characteristics of the transition state ensemble and which contacts are formed when the system is sampling this transition, you tend to get pretty good agreement between most naturally occurring proteins and what these models will tell you. And when you look at synthetic proteins, then it tends to break down. Artificial proteins tend to do more poorly, and that's probably because when we design proteins, they're not as funneled as nature has figured out through evolution. So another thing that you might see in the literature and talks is people talking about multi-basin models. So I'll just walk you through one type of a multi-basin coarse-grained model. So this is still structure-based, but now in this example, this is adenylate kinase. I'm showing 2 structures where this lid domain and NMP domain undergo about a 5 to 10 angstrom conformational change. And so we would like to adopt this approach to look at conformational changes as well, and there are some sort of straightforward ways of doing that. And I'll just give you an example of what we have done in the past. This is not the only way to do it. There are actually many approaches that are out there. So essentially what you need to do is when you are looking at your contact map, you need to decide what contact map would you like to use and what distances should you use, because now it's not uniquely defined. You don't have a single structure that could define a minimum. You have 2, and you need to think, how do you want to combine those terms into a single force field so that the system naturally will interconvert between these 2 endpoints. So when you hear multi-basin coarse-grained structure-based model, it's really usually just saying the contact map is not simply from one structure. That's oftentimes what is done. So oftentimes it might require you to construct a few variants. In this case of adenylate kinase, we had some information about the balance of configurations of open and closed states. There was some fluorescence data that was available. So we constructed various combinations of these maps to see could we get the overall picture of the conformational change to be consistent with what we knew, and then we started using that model to actually study the dynamics and how the system responds to various perturbations. So when you're constructing a contact map now, you have 2 maps, so you need to actually calculate those maps twice, one for one structure, one for the other, and then you need to start comparing these 2 maps and deciding what do you want to keep and what do you want to exclude, and what sort of physical interpretation does that combination give you. So what we did is we said, well, we have an open configuration in a closed configuration, let's calculate both of our contact maps, and we'll simply include all of our open contacts as defined by the open configuration. So every contact will favor this open configuration. And then we said, well, if we look at contacts that are only formed when the system is closed, if those contacts are completely broken in the open configuration, we will also include those. And those contacts we consider to be related to ligand binding. In this case, it closes when the ligand binds, so this was an effective way of accounting for this ligand binding induced conformational change. So it's not that these are direct interactions in nature, they're actually here describing an effect of a ligand. So what we did is we took all of these contacts and we give them a strength of, say, 1 as a starting point, and then we put in these contacts and we vary how strong those contacts are. And as we make these contacts stronger, we would anticipate that the system would spend more of the time being closed than open. Although, you may not see that as the case if you weren't careful with how you constructed your maps. Just a schematic of what things look like. If you want to go down this road, we will often take, in this case, the closed contacts, and we'll calculate those atom-atom distances in the two conformations and see if there's a nice separation of the population of these contacts. So in this case, there were many contacts that are along this line of slope 1, which means even though they were only identified in the closed configuration, those atom pairs have about the same distance in both conformations. So we wouldn't say that those contacts are really uniquely closed. It's just that our algorithm for catching contacts didn't identify them in one structure, but it did in the other. So what we did is we said, well, let's just ignore these for now and only look at these contacts that are very clearly broken when the system is open. And we call these our Q unique or Q ligand contacts because that was our effect of the ligand, was to put in these very uniquely closed interactions. And when you do that, there's no guarantee you're going to see the other state. It could be that due to the competition of these interactions, you'll get some distorted system that isn't closed and isn't open. There's always that possibility, so you do need to check. It's not, even though I'm giving you sort of a recipe for trying things, it doesn't mean it always works. Right? And if it doesn't work, you can probably learn something about the system. Why doesn't that description, why isn't that enough information for you to recover at least the endpoints of this system? So as I said, things that we looked at were, say, distribution of conformations between open and closed. There were some measures of inner residue distances. There were some B factors from crystallography. So we had a variety of experimental pieces of information we could compare against to say, is this model at least in the right ballpark? Is the molecule fluctuating with the right scale? Can it interconvert? And does it actually go between the endpoints, or does it just go to some sort of strange intermediates? So this is an example of what we saw. As you strengthen your closed contacts, the population becomes more and more closed. This is just one of the domains. When those closed contacts are really weak, it basically sits in this open configuration all the time. Okay. And then in this model, we looked at both domains and how they were moving. This is fully open, this is fully closed, and what became apparent was that you get this very pronounced intermediate where one of the domains closes and the other one doesn't. And you can start, what you can start to do is start asking, well, what is the interaction between these two domains? And what we found from that by closing one domain, it actually opens up an interface for the second one, which helps stabilize the closure of the second domain. So there seemed to be this sort of dependence of one domain on the other domain. And I won't go into why we thought that was useful, but we found that to be interesting, and we did a lot of perturbations to try to understand what biological role that could that have. So even though we only put in information about the endpoints, we get this naturally occurring intermediate with this model, which is similar to what I was showing you with the ribosome, where we used a single basin model and we get all sorts of intermediates there as well. So just because you define the endpoints doesn't mean that's all you're going to get. There are other approaches to using multi-basin models, and with those, sometimes you can explicitly define the endpoints and only the endpoints are minima, in which case that's all you're going to get, and you will always force it to be two-state. But in this case, we're not putting that restraint on the construction of the potential. So these are all effective terms. So we know that when the ligand's bound, the domains are closed. So the question was, if you only include those close contacts, and you have to sort of play around with how strong they are, is that enough for the system to be closed? Or it could have been that when you put in those close contacts, you only get this state, but you don't actually get the fully closed state of the system. Maybe there just isn't enough information in those contacts to even get there. So it's a heuristic sort of approach. You're tuning the overall phenomenological properties, so then you can start adding perturbations afterwards. That's sort of the style of how these models are used. Oh, sometimes you have, it depends on the system. You can have single molecule FRET data. Sometimes you'll have NMRs, sometimes B factors for fluctuations at the endpoints. Fluorescence tends to give you a nice changes in population, and it'll give you relative distances of certain regions. So it's really specific to the system that you're looking at. In the case of the ribosome, we had single molecule FRET data. We had crystallographic B factors. We had some explicit solvent simulations. So we had a lot of pieces to make sure that we weren't seeing anything that was unreasonable, that the overall scale of the fluctuations was appropriately described. Right? So that's part of the homework. Is these are certainly not models that are one size fits all and just go. And I was telling someone earlier that we have a listserv where people send their questions, and sometimes they say, "Oh, can I do this?" And on the one hand, you can do anything you want, but you have to first ask why. What would you like to learn about your system with these models? They're not. It's a sort of a philosophical difference from all-atom explicit solvent simulations, where you say, well, if you define all of the atomic interactions, that just gives you a landscape, and it's a matter of waiting for the system to sample it, and you simply report what happened, right? Here, we have to include so much information up front that there's always a possibility that you put in so much information that your answer is predetermined. So you need to step back and think, what you're interested in, does that depend on these parameters or not? So if you have specific systems, I'm always happy to talk about your specific systems and brainstorm, but you really do have to give a lot of thought to what could you learn for the application. Like I said earlier, in the ribosome case, we were looking for steric factors and the effect of flexibility. And those were two contributions that we were trying to identify or seeing if they were significant. So that was why we applied that model. And if you were looking at changes in hydrogen bonding energies, this model would be completely inappropriate because you're basically having to put those numbers in up front. Yes, of course. Deferred back. So for well, you do need structural information to define the force field in the first place. So if you have no structural information, that sort of takes structure. It takes the structure out of structure-based, right? So for intrinsically disordered proteins, I have not tried to venture off in that direction, because essentially, I mean, the way I would approach it would be maybe a coarse-grained model, but you would have to think about other parameters for degrees of flexibility based on residues. For example, maybe the contacts would be structure-based if you're looking at, say, fibril formation. You have lots of small peptides, and you know what the fibril looks like, and you want to ask, as you change properties of the peptide, how does that affect formation of the fibril? That would be a perturbation and an effect due to that perturbation. But you know that it forms the fibril up front. So you would basically be encoding the fibril and then asking how does that formation get perturbed by other factors, like changes in dihedral energies or changes in specific side chains. So in those cases, maybe, but again, it's very, if it's intrinsically disordered, it sort of takes the structure out of structure-based, so I would be, careful. You'd want to be very thoughtful in what you're doing exactly. Right. Well, that would be a signature that the helix is marginally stable, so you could always perturb the parameters of that region and just make it a less stable region. You still have to think what the question is you would want to address with that model. You can always just play around with it and make it do things. So what the philosophy I take is I try to make it do things to some extent because I want to ask how does it respond to perturbations, right? So if you get the overall picture, whatever information you have, and you have that general picture accounted for, then you start adding perturbations to the system. How does the dynamics change? That's sort of the philosophy of how I approach using these models, typically, rather than we're trying to get the exact balance between ordered and disordered states. Maybe we're just looking at the shift when you put in some other perturbation, when a ligand binds, as you change temperature, or if you have an effective pH, for example, electrostatic effects. You can start adding these layers in and ask how does it perturb the picture from that initial view that is roughly consistent with what you do know already. So again, it's a very different perspective from conventional explicit solvent models where you're really trying to get an exact answer. I look at these more as being useful for understanding relationships and trends, and exploring potential relationships given a variety of assumptions. Right? So those are the coarse-grained models. The all-atom models are actually formulated the same way. The only difference is that we explicitly represent every heavy atom by default. You can also extend that and include every hydrogen if you are so inclined to do so. So it's okay. You can put whatever resolution you want. In the all-atom models, the force field is exactly the same. This improper term, improper dihedrals and planar dihedrals do come in because you have rings and because you have chirality that can be lost because you've neglected a hydrogen, for example. So you might need to put in improper dihedrals. But other than that, the force field is actually has all of the same elements, except now these R naughts are no longer distances between C alpha atoms. They're actually the chemical bonds in the system. And in this case, you might want to relax the distances and these angles because now you're actually talking about real chemical bonds, chemical bond angles, as opposed to the C alpha model where you're talking about C alpha, C alpha, or C alpha, C alpha, C alpha angles, right? So it makes more sense to really put specific angles, specific distances in the coarse-grained variations, where in the all-atom version, you could just take, say, the amber backbone parameters for bonds and bond angles, and that's not going to affect any of the results with this model, assuming you're using a structure that is reasonable. And this comes back to your sidechain question. So if you really wanted to know how does sidechain composition play a role, well then you would probably want to use a model that actually has sidechains represented. Otherwise it's going to be very difficult to look at sidechain dynamics in an implicit fashion. So again, depending on the question, you need to pick the model that is most suitable. And these all fall within the same class. This lattice model, the off-lattice C-alpha model, the all-atom model, they're all constructed in the same way. They're just different resolutions, right? Spatial resolutions. It depends on what you're looking at. There are some proteins, like protein L and protein G, where the packing of the side chains can actually change the mechanism substantially. So one of the early all-atom structure-based model papers with Jose, Angel, and Cecilia Clementi, they actually saw very different folding dynamics using an all-atom model of this form, even though the tertiary structure is almost identical between these two proteins. So if you use a C-alpha model, the contact maps at a residue-residue level look very similar. The backbone geometry looks very similar. So essentially, you can't distinguish between protein L and protein G. But experimentally, they saw very different kinetics of folding. And when the side chains are represented, then the sterics from the side chains can actually start shifting things all over the place. So right, so if you're trying to compare two systems that have the same tertiary structure, then a coarse-grained model will just basically give you the same answer for both. might really want to look at these all-atom models. In the case, I mean, of course, my favorite example is the ribosome. If you were to coarse grain the ribosome and you put one bead for every residue, those steric barriers that I was talking about earlier, those are very finely defined. And you start putting spheres in instead of actually the atomic geometry, then some of the barriers get amplified dramatically because you're putting a hard wall, essentially, on these big beads, or you completely miss them because the beads are not positioned at exactly the right spot. Because you're talking about a lot of these steric barriers, if you move things at 2 to 3 angstroms, you can completely eliminate the barrier, or make a barrier go up by like 50 kT because atoms are basically just on top of one another. So if you're really looking at fine steric effects for your problem, then these would be the models that you would want to use. You could also use a combination where maybe you only have the backbone in atomic resolution and the side chains are implicit because the side chains are all packed during what you want to look at. Right? And you could reduce some degrees of freedom. So I'll just give a couple of brief examples of things you can do with the all-atom versions of these models, just to give you sort of an idea of things you can play around with. I should say that these models are actually very cheap in general. So if you're looking at anything that's less than a few hundred residues, you could probably do it on a desktop without much work and get native basin fluctuations in a matter of hours without a problem. If you're looking at a large-scale conformational change, it could still take a while. So this was a case from José's group where Heiko was working with Rachel over in Israel, and they had experimental data showing that the electrochemical potential of ferredoxin changed when you perturbed this loop, which was at the opposite side of the protein from the active site. And these are the two crystal structures. So the tertiary structure of these two proteins is basically identical. There was nothing really distinguishably different between them. And the only difference was that 2 residues were removed in this far loop, and they were trying to figure out, well, how could this occur? Right? There was no obvious conformational change. There was no change in the binding pocket. So what Heiko did is he actually just constructed a structure-based model and removed these 2 residues. So by construction, the model is identical energetically, except for this truncation of this loop. So you could think of it as you are pinching this helix. And what he actually found was that if you pinch this helix, the system naturally starts to fluctuate more on the other side. So this network of interactions, if you pull on one side, it naturally opens up on the other side, just due to the connectivity of the interactions. It had nothing to do with sophisticated electrostatics or hydrogen bonding or anything like that. It's just that it's connected and the network responds. And those calculations, again, could be done. Of course, the first round didn't necessarily work properly. But in the end, these calculations would require like an afternoon on a desktop these days. So you can start to really play with ideas and look at this idea of how can perturbations have a role in your system. And so this actually made a lot of sense and was able to rationalize all of the experimental data that they've been sitting on for years. Because everything they did crystallography, thought they'd see a change, saw no change. They see electrochemical changes, great, but there was no signature as to why. Another example of something you can do is now that we've gone to these all-atom resolution, the all-atom resolution of these models, is you're not limited to proteins, those C-alpha models. Obviously, if you don't have C-alpha atoms, you don't have a C-alpha model, right? So you can look at RNA, you can look at DNA, you can look at ligands, but it's all within this structure-based framework. So everything you're including is still structure-based. It's all defined based on a particular configuration. It's just now it's RNA, or it's a ligand, or it's DNA, or it's a prosthetic group, or a modification, or whatever it is that you'd like to look at. So when we were getting these models going, there's interest in the SAM riboswitch. There's this 94 residue RNA, and it binds S-adenosylmethionine right there in the binding pocket. And the question was, could we get an idea using these models of how this ligand binding might affect the folding process? And so what we did is we took these models and we did kinetic simulations as well as thermodynamic simulations, just probing the entire folding process of this molecule, and we included it without this ligand. And you get some ordering of your helices, and then we could repeat it with the ligand present. And this purple line is just showing when the ligand binds during this process, and we also saw that there was this effect in a certain region. So ligand binding actually destabilized part of the system. It did what's called backtracking before the system fully folded. And there was also this large effect. It's sort of hard to see when you look at these two, but if you see this is shifted from 0.8 to 0.6, that just means this region of the RNA was folding earlier in the process. And that region was this terminal helix, so it wasn't completely surprising that you would have an effect on this helix. But what we thought about was that this is the terminal helix where you have two tails coming together. Without the ligand, it's very much the last thing that happens, because there's this entropic penalty to fold this tail. And if you look at a lot of the riboswitch literature, people say all the helices fold, and they don't even think about non-local helices being entropically disfavored. It's just all helices are folded and then something else happens, and that's sort of the working model. And what this suggested was that, well, that entropic signature is very strong, so you would expect it to be very late that this folds. However, when the ligand is present, it starts to fold significantly earlier. So it does actually change the accessible pathways of the folding process. And it was a different picture from what people were talking about at the time. So it's not that this is the perfect picture, but it's a very sensible signature and influence on the overall folding dynamics for the system. Now, as far as things you might want to do, if you wanted to look at ligands, this is sort of elaborate, but we had a bath of ligands, right? So you don't have to say this ligand binds at this time and you force it to bind. You can actually put in arbitrary copies of the ligand where all of those ligands are competing for the same binding site. So this SAM that binds is random, which one it is. Once one binds, it occludes the binding site and nothing else can bind, but you can actually look at the effects of conformational changes with a bath of ligands and simulate the process hundreds of times under various conditions. So if you have ideas of how a system works, ideally you should be able to take those ideas, formulate them into an energetic description and say, does that energetic description give you, anything reasonable? Or is that energetic description just giving you complete nonsense, which would tell you that your ideas are probably not right? If you want to think of a real purely physical physicist way of looking at something, you would start with a Hamiltonian and say, here are the principles that are important, and then we can derive everything. Here we're saying, well, is this picture okay? Well, then we should get answers that are consistent with pieces of information that we know. And if it doesn't, then we've missed something. We need to go back to the drawing board and think something else might be important during that given process. So you can do examples like this as well, where you have baths of ligands and you have spontaneous binding. And again, these are all structure-based models. Okay, so nothing I've shown you has any electrostatics. These are all purely defined based on known structures. These are just different variants of the same theme. Okay. For those of you who are interested in the practical side, tomorrow we'll go through an introduction to actually performing a coarse-grained model. On Friday we'll go through the all-atom models and look at some other considerations. So all these models are freely available through a web tool that we've made. If you have a structure, it will give you the force field, and then you can just use GROMACS to simulate it, or NAMD or AMBER, actually. Now they're all supported. So you pick your favorite MD package. This tool will give you the force field, and then you can start doing all the normal MD types of calculations, enhanced sampling, whatever you would like, to that model, with that model. So I'll thank Jeff. Jeff has really built a lot of these tools with me through the years, but those were all good questions. So that's the introduction to what these models can do and sort of the range of things that one might want to do with them, but all within this same overall philosophical framework for how to study a system. So we kept it about 45 minutes, so dinner won't be delayed for anyone. If anybody wants to ask more questions, I'm obviously around, so feel free. So I'll see you guys tomorrow.